So I think we'll go ahead and get started again. We had a great start this morning with our speakers, but we're going to put a little bit of pressure on our next three speakers. In this session, we're looking at tissue chips and really talking about state of development. And the idea here is to highlight some of the issues that we just talked about, but with some given organ systems that are under development. And we've got three examples of that coming up. Our hope is that these individuals who are all NIEHS grantees and who are working on these particular systems will give us some opportunities to really interrogate the systems a little bit and understand them and use them as some examples as we have our discussion later on today and tomorrow. We're going to ask the speakers to spend about 25 minutes total, and the yellow light will come on at 20 minutes, just so that you know when you're running out of time. This is the only way we'll be able to stay on time today, and we have to bring someone in by phone and webcast at exactly 1:15. So without further ado, our next speaker is Dr. Julie Kim from Northwestern University. Julie is Associate Professor of Obstetrics and Gynecology and Reproductive Biology Research at Northwestern University's Feinberg School of Medicine. Julie.
Thank you. Thank you for inviting us to speak about our system in the female reproductive tract. I'm very happy that it is one of the organ systems that you've decided to highlight here. This is really work, it is a team effort done by multiple reproductive biologists in our division. Teresa Woodruff is the PI of the grant mechanism that we have currently, the UH2 and the UH3. And I will tell you about the reproductive tract specific organ units that we have that make up the female reproductive tract. And I usually like to start a talk with a joke, but given that I am going to talk about the female reproductive tract and I am at the National Academy of Sciences, I decided to behave myself and skip the joke. So let's go right into the talk. I'm just going to give you a basic overview of why the female reproductive tract is important to include on any kind of chip system or in itself study in a chip format. I'm going to talk to you a little bit about the building and the testing of our reproductive systems in vitro, and then the system that we are using to integrate the FemKUBE. And then I'm going to put out some questions and challenges about going beyond the FemKUBE and looking at disease modeling that require effects of hormones, female reproductive hormones. And so if you go back to middle school, I'm sure you've learned the female reproductive tract to be an integrated organ system comprised of distinct functional units. Its main purpose is to reproduce the human species by providing half of the genetic material and a place for gametes to fertilize and to develop. And so this is where it all begins. This is where you and I all began, right in the fallopian tube. And we can all pretty strongly agree that without the female reproductive tract, mankind will cease to exist. And so that is a very strong argument or a reason to make sure that the female reproduct is preserved and protected. For obvious reasons, research in the female reproductive tract is limited, and that has been quite a challenge in studying the reproductive tract and its effects on the human body. The most obvious one is the ethical issues associated with experimentation of women during the reproductive age, as well as in pregnancy. And interestingly, reproduction in the species, different species, is very different. And there is no good model for human reproduction except for the human model. So such differences in species variation, it stems from estrous cycle, number of follicles that ovulate, the implantation process, the type of placentation, the length of gestation. I mean, these are all things that differ from humans. And so that is a good reason for us to really go back to the human to study human reproduction. Now, not only are the structural components essential for reproduction, but the key hormones that drive this whole process are the ovarian hormones, mainly estrogen and progesterone, as well as other hormones that are produced in the ovary. And this dynamically changing environment, and I'll show you it in another slide, doesn't affect just the reproductive tract, but it affects the whole body. And it affects organs such as the heart, the bone, the liver, the brain, and the gastrointestinal system. So you can imagine that when you start testing certain compounds or drugs, that it's going to be essential to have that layer of reproductive hormones on top of your compounds. And so for these reasons, a lot of the preclinical and clinical research that has been done have been done in males. And when a graduate student has asked one of her professors why this is so, he just simply said, we don't study females. Their menstrual cycles would mess up our results. That's because the female is very complex, and I say that in a biological term. It's not only does a woman have menstrual cycles in a certain span in her lifespan, about 35 to 40 years, but there are also pre and post menstrual menopausal states, and when you're pregnant, the levels of those hormones skyrocket. So we are constantly being bombarded or devoid of these hormones, and that could be the underlying reason why more women express higher rates of anxiety and dementia, or men have a greater ponderance for likelihood of getting autism or Parkinson's disease. The reasons for these sex differences are not quite clear, but it's probably due to the hormones. And just a thought, in the past decade, 8 out of the 10 drugs that were pulled from the market for being unsafe were found to have more serious side effects in women than in men. And so you can imagine why then it is important to have a reproductive system, or something at least, to produce those reproductive hormones in a platform where we can test early drugs, reduce that sex bias in the pipeline, and increase the safety of drugs. There are sex-specific drug metabolism capabilities. We can also study the gestating uterus for drug development. We can also use such models for contraceptive design and targets, and to really show that it's not really causing an abortion per se, but preventing ovulation. We can also use this system to study vaccines, and I'll show you later on, potentially use it for studying mechanisms of cancer prevention and therapy. And so on the left, you see reproductive tract of the primate. It is comprised of the ovary, the tubes, the uterus, the cervix, and the vagina. And we have decided to model this in a physiological platform in collaboration with the team at MIT and Draper. And so I'll get into this more later, but what we have are just these modules that have the different organ systems that are going to be integrated together with a microfluidic platform. And as I said before, during the menstrual cycle, there are so many changes that occur in a woman's body, starting from temperature all the way to the levels of ovarian hormones, the levels of gonadotropin hormones, what that does to the endometrium during the menstrual cycle, the changes that occur in the ovary with folliculogenesis, etc. And so it's not really a static exposure. It's always moving. It's constantly changing. And it's this dynamic fluctuation that are causing those biological responses. And so it's very difficult to study this in a static culture system. And so we feel that the microfluidic system is perfect for studying such a situation. And so our group has, first of all, in order to tackle this problem, has decided to take each of the organ systems separately. So Teresa Woodruff is in charge of the follicle, the ovary, and programming those follicles to make the hormones that mimic the menstrual cycle. Joanna Burdett is studying the fallopian tube and the biological responses that occur during the menstrual cycle to ovarian hormones. I'm leading the team that I'm studying the uterus and the cervix. And what we have done in vitro is to basically mimic the in vivo hormonal profile in a stepwise fashion, where we'll treat the cultures for 7 days with estradiol only, and then increase that level of estradiol to 1 nanomolar, and then add in progesterone at day 14, etc., increase progesterone levels, and then at the end of the menstrual cycle for about 2 days, withdraw those hormones so the tissues are devoid of the hormones. And so this is how we kind of recapitulate that changing hormonal profile in vitro in our static cultures. So if we look at the ovary, and each of these reproductive units, we've called them cubes. And so the ovary cube, here you see a human follicle encapsulated in an alginate bead. And so it is in a three-dimensional structure. And so we'll take a human follicle from a secondary follicle, and you see that in vitro we are able to mature it so that it eventually develops into a mature follicle. Here you can see an antrum with the oocyte. And as well, it can release hormonal profiles in vitro, as you would see in vivo. So by the addition of FSH and HCG, we can mimic the expression and production of hormones like estradiol, progesterone, inhibin A, inhibin B, AMH, and activin A. And you can see that, for instance, for estradiol, you see that rise in estradiol during the follicular phase, and then you add HCG to mimic ovulation, and then you see a decrease. Whereas with progesterone, you start seeing that increase of progesterone production after the addition of HCG, which is around the time of ovulation. So this is all great for using human follicles in vitro, but we know that we cannot always go to the surgery room and get human follicles whenever we need them. There are some limitations in studying human follicles. And so what Teresa's group did was she took mouse follicles. Remember, mice have an estrous cycle about 4 days. It's very, very different from the human. But she took mouse follicles and similarly encapsulated them in alginate beads, and she was able to phenocopy the human menstrual cycle profile with mouse follicles. And so just by adding in the FSH and HCG in a certain pattern, what you see here is a very nice increase in estradiol by those mouse follicles, followed by an increase in progesterone during the latter half after the addition of HCG. And so we were very, very happy to see this phenocopy the human menstrual cycle. And so with this, she is able to then provide a system that provides a source for ovarian hormones, as well as provide a model to test compounds on follicle survival or follicular health. And so I'm going to give you just a couple of examples of what she's done with this system. These are some of the parameters that are measured to assess follicular health, such as morphology, their ability to develop, etc. And so with this system, she has tested 2 chemotherapeutics that have known adverse reproductive effects of follicles in vitro. And you can see that cyclophosphamide and cisplatin with increasing doses do decrease survival of follicles. And this is in a dose-responsive and time-dependent manner. This is another compound that was used in the 2010 oil spill. where the Corexit 9500 is actually an oil dispersant that they put on the oil so that the oil can disperse and then eventually get degraded by bacteria. There were no known, it was not tested for any kind of toxic effects on humans or animals, but it was just used on the oil spill. So here is an example of how the follicle system can be used to assess the toxicity of compounds that have unknown effects in vivo in an in vitro system. And you can see here with Corexit that increasing doses of Corexit does indeed affect the survival and the quality of those follicles in a dose-dependent as well as a time-dependent manner. And just as a nice control, we also have exposure of these follicles to an analgesic that is commonly used called nalbuphine, and it does not affect follicle quality or morphology or development. So next I'm going to talk about the tube cube. This is the fallopian tube that we get from human fallopian tubes from after surgery. And what we do, we just peel off that epithelial layer and then we culture that in transwell membrane culture systems. And these systems do survive for prolonged periods of time throughout the whole menstrual cycle. And one of the characteristics that we are able to study from fallopian tube cells is actual cilia beating. And this cilia beating can be visualized in this in vitro culture system very nicely, and it can also be quantified. And so you can see here that these are actual beats per minute. And we see that the addition of estradiol, as well as estrogen and progesterone, they do regulate that beating of cilia in these cultures. And so you can see how the hormones are affecting the physiological function of this tissue. We are also able to measure secreted factors via the fallopian tube. The important ones are oviductal glycoprotein 1, which is indeed regulated by estradiol, as well as estradiol and progesterone. And also IGF-1, which is increased with estradiol during the initial 14 days of culture, and then that is diminished in response to progesterone. And so we have some secreted factors we can measure, and we have some physiological function as well. And I just want to show you this very interesting slide where we added testosterone into the system to see what it would do to cilia beating as well as secretion of oviductal glycoprotein 1 and IGF-1. And you can see that OVGP1 does increase when testosterone is present. It decreases level of IGF-1, and strikingly, you can see that the cilia beating here in response to estradiol, but as soon as you add in testosterone, that cilia beating does not occur. And so it's a very nice marker for looking at exogenous compounds to test physiological function. Now, I have been charged with developing the uterus in vitro, and I have to say that is the hardest organ system to develop. It is comprised, the uterus is comprised of many different cell types. The endometrium is the lining where the embryo first implants, and it is made up of stroma, the epithelial cells, and then that's surrounded by a nice big muscle layer called the myometrium. And so we decided to kind of tease this apart, and we've tried multiple different scaffold compounds to grow these in, and we've chosen a commercially available polystyrene scaffold that is shown here to grow epithelial and stromal cells of the endometrium together, and then transwell that on top of myometrial cells that are grown on this layer. And so you can see that the cells do grow in a 3D-like fashion, and we've been testing the system for secreted compounds to determine what are the secreted factors that are being released by this system. And basically, just to summarize a lot of data, what we do see, what we have identified are some cytokines and some growth factors that are preferentially secreted during either the proliferative phase of our in vitro hormone system or during the secretory phase when progesterone is dominant. And so these are some of the factors that we've identified using a Luminex multiplex assay. And the same thing with the cervix and the FemKUBE. We've grown the cervix in these scaffolds, and you see them bunched up together, growing in a nice 3D-like structure. And one thing that the cervix does really nicely, the endocervix, is it produces mucus. And so we are able to see that the estrogen and the progesterone do induce the production of mucus. MUC16 here is stained. We also use different stains that stain different like neutral mucins, as you can see in red here, as well as some acidic mucins, as you can see in blue. And so really nicely, we do see these cervix cells in vitro during this menstrual cycle period producing mucus. And again, with the Luminex system, we also have been able to identify some secreted factors that are expressed preferentially during the proliferative phase versus the secretory phase. And so we can use these as markers later on when we're testing different things. And so as we have characterized the static systems of each of the reproductive units, our goal is to really put them together and integrate them into one unit. And so we're going to integrate the follicle with the tube, with the uterus and the cervix, and expose them to in vitro menstrual cycle hormonal profiles. And this is done really in collaboration with engineers at MIT, headed by Linda Griffith, as well as Jeff Bornstein from Draper Labs. And I'm just going to show you just very briefly the platform that they have developed for us. Again, it's like a plug and play. These are basically individual modules that you can plug in. Here in the diamond-shaped modules, that you can see it closer here, are where the follicles are going to be harbored. We have a place for 4 follicles to sit there, and then fluid is going to flow through this module and then into one of the chambers where we will put in our target tissues, whether it be the tube, the uterus, or the cervix. And here is just a little example of the flow through of a dye through this module that will be harboring the follicles. And you can see that the flow is pretty even and goes through at a certain pace, certain microliters per minute in this system. And so this is what it looks like in real life. It is still being tested for that hormonal profile. We have done a couple of experiments to determine the concentrations of estrogen, progesterone, and activin and inhibin within this microfluidic system, and our initial studies are definitely showing very similar profiles as aesthetic cultures, which we're very, very excited about. And so with this system, you can imagine that how it can be applied to multiple different systems. For instance, we can study the follicle with one target tissue at a time, or we can put the follicle together with the tube, the uterus, and cervix, and then study all of the downstream effects linking all of these units together. And we're pretty flexible. We are in the process of building these 2 different platforms. And we are also able to add in the test compounds directly onto the follicle module, or if we want to determine what effect those compounds have on the other tubes without affecting the follicle, because we still do want to see that estrogen and progesterone being produced, then we can add it downstream and not have the follicle exposed, etc. So there's a lot of flexibility here that we're working with. and I was kind of alluding to before, we can study other disease models, specifically cancer, of tissues that are from hormonally derived tissues, for instance, endometrial, ovarian, and breast cancers. The role of hormones in these cancers are very important, but in terms of its initiation, but in terms of what it does to the actual cancer, it still remains unclear. And so you can imagine that the follicle as a source of hormones can be linked to the diseased tissues, the tumor, and then we can study how that affects potentially metastasis, either to the mesothelium or to bone, for instance. And we can then test a lot of different compounds to determine how that movement is affected. We can also study, use these microphysiological systems to study diseases such as leiomyomas or other benign diseases that are hormonally responsive. So for instance, hooking on a follicle module with leiomyoma, as well as an endometrium, let's say. It is very well known that progesterone, for instance, promotes growth of leiomyomas, but then progesterone is important to keep that endometrium from proliferating too much. So if you were to add in an antiprogestins, let's say, to the female reproductive tract, you can have bimodal effects where it can definitely decrease growth of leiomyomas, but what will it do to the endometrium? And so this is a nice in vitro platform where we can study such cases. And finally, this is another usage that we are interested in as well, in collaboration of the HIV researchers at Northwestern, is to really look at HIV transmission in the female reproductive tract. And the researcher that we are working with, Thomas Hope, is interested in looking at cervical mucus and how antibodies to HIV can trap HIV at that cervical mucus. But if you have mucus that changes throughout the menstrual cycle, it's going to be very hard to study in one system. And so this is a very nice platform where you can study the flow going this way and then HIV trying to get in this way. And so this is another potential application. And then above and beyond that, eventually we can look at pregnancy modeling by putting in a placenta. We'd definitely like to hook it up to other systems like the liver to study drug metabolism and how ovarian hormones can affect drug metabolism. How does it affect the bone? And then eventually it's going to be very important to include the male reproductive tract as well, what we're going to coin as DudeCUBE. And so I'm just going to finish here with two specific challenges. I'm sure there are more, but we can talk about two particularly. It's the human tissue availability. Unless you have a system that we have at Northwestern where we have a collaboration of doctors, nurses, research coordinators, so many people involved to allow us to get that tissue from the OR, then it's going to be very difficult to study these systems. And so we are trying to develop some of these tissues using iPS cells. This has never been done before in the reproductive tract, so it's going to be quite a challenge, but I think we can do it. And so that at least will give us a nice consistent pool of cells or a good cell source for us to study such tissues in vitro. Optimizing the 3D cultures. We have a basic idea now of how these cells or tissues that form on these platforms behave, but I think we can do a little better. So currently we would like to, or we are pursuing a different kind of scaffold, and I think it was mentioned earlier this morning, about decellularized tissues as scaffold, where we can actually just take cervix or uterus or the tube or what have you, decellularize that. So all you're left with is the ECM and the scaffolding for us to use as a scaffold to then eventually seed in with the cells of interest later on. And our initial experiments with the follicle cell as well as the tube are very, very interesting and very encouraging. We see that the cells are kind of honing in and organizing in a way where they kind of know where to go and form certain structures. And so we're hoping that this will provide us with a better scaffold system. And of course, we cannot do this alone. This takes a whole team of people. This is our FemKUBE team, and we are collaborating with a lot of different researchers from MIT as well as Draper, and this is the gang from Northwestern. And with that, I'll take any questions. All right.
This is quick. The question is that you showed some beautiful results about maturing the follicle, which is like the functional unit that you want to ask the questions about. But I wasn't clear in the uterus and the cervix. Are those like primary samples in culture, or are they cells? Does the tissue self-assemble? What's going on there?
So we do take tissue from the OR and we digest the tissue, so we get single cell suspensions. And one of the limitations is that we don't have enough of that material to make as many as we want. So we do grow them in vitro in 2D to expand them in culture to get enough cell numbers, and then we passage that right onto those scaffolds. Does that make sense?
Yeah. Hi, David Gerhold, NIH NCATS. And so it occurs to me when you're talking about the sex differences in response to drugs, that the response to the somatic tissues is generally dominated not by testosterone and estrogen, but by the growth hormone cycles that they drive. And so have you thought about replicating that system so that everybody else's model systems can be differentiated to resemble male and female cycles?
So are you talking about the peptide hormones that are also being released by the ovary?
Well, I'm thinking actually about the human growth hormone cycles that are driven by the hypothalamus and the pituitary axis.
Okay, right. So that would be really, really nice to have that part of the brain on a chip as well. And we were kind of thinking of having that at the beginning, but I think for now, to make everything simple, we'll add in those exogenous hormones, or just have that follicle produce whatever it needs.
Or could you just make a system that would derive cycles or pump cycles in the human growth hormone?
Yes, absolutely. Right. Yes. Thanks.